We are very pleased to have Clara Thiel with us. She is a lecturer of biology and forest ecology and the coordinator of biology internships at Frostburg State University. In addition, she curates the Frostburg State University Herbarium. She is from Clarke County, Virginia, and earned her bachelor's degree from James Madison University in Harrisonburg, Virginia. She earned her master's degree in applied ecology and conservation biology from Frostburg State University and currently teaches courses in plant systematics, economics, and field identification. Her current research focuses on insular ecosystems and edaphic plant communities throughout the Mid-Atlantic region. Thank you, Clara, for joining us.
Thank you so much, Judy, for your introduction, and Maryland Native Plant Society for having me for another talk. I am very happy to be chatting with you guys this evening about the Frostburg State University Herbarium, or our dried plant collection. So that just kind of jumps right into my presentation. First, I just want to go over what is an herbarium and what is a plant specimen. And a herbarium is just like a library of dried plant specimens that have been vouchered. That is the term we use to describe the collecting of a biological specimen. And it serves as a repository for these specimens that are extremely rich in ecological data. We're able to mine a lot of information from dried plant specimens, not just about plant morphology, but also about its ecology, and many aspects of that, which I'll get into further down the line. But primarily, an herbarium serves as a few main primary sources. And the first is as an identification reference. Herbaria store what we refer to as a type specimen. A type is a specimen that has been established or recognized as the official physical reference for a species. So this type specimen here is an example of a maidenhair fern special variety that is known from the Appalachian region that was first described in Ohio. So this is in a repository in Ohio, and this serves as the official type specimen for that variety of maidenhair fern. And having that physical specimen also allows us to track changes in authorship of species over time. And as you would imagine, the describing of variants and subspecies, having these physical references are really important. Another primary purpose of herbaria is to serve as a validation source to validate that a species was indeed documented. So recently, back in 2021, we got a new Flora of Maryland, the Vascular Plants of Maryland. Wes Knapp and Rob Noxie put this together. And it took a lot of work investigating all of these, many of them true and some of them not true, claims of plants established in certain areas. And essentially what they did is very closely examine that original list or floristic inventory of plants that occur in Maryland and essentially validate every single one of those claims with a official herbarium specimen to prove that it was indeed documented in Maryland. So they're very important to those inventory studies of diversity within a region or space. And then in addition to that, they can also serve as documentation of a occurrence that, say, is priority for survey or conservation initiatives. So some cool specimens like the Cypripedium pubescens, or yellow lady slipper, was collected way back in 1940 in Garrett County, Maryland. And we don't have a lot of information from the label. All they say is Big Savage Mountain. But this is, hard evidence that this regionally uncommon plant did occur in this, at least, locality in the 40s. And then lastly, the last main use of herbaria are a source for species discovery. So within these herbarium specimens, you could imagine that there are many inconspicuous species, think sedges, that are really hard to recognize or look superficially very similar to other taxa. So when describing a new species, taxonomists will often reach out to other herbaria and request loans of specimens of a certain species that they're studying to help with whatever research they're focusing on. Again, this is really important for regional variants or subspecies. And then especially with recent advancements, we're able to also use herbarium specimens as sources of genetic material, especially with our new methods where much less tissue is required. You can take a small leaf sample from an herbarium specimen, even over 100 years old, and be able to yield some significant chemical or genetic analyses from that sample. So, just to go over the parts of a herbarium specimen, again, this is a dried, pressed plant that is put into a plant press. Essentially, you're taking a 3D plant and making it 2D. So the idea is to spread out the morphological features as best as you can, so you can realistically represent both the growth habit as well as how the leaves were arranged, while also displaying both the top and bottom of the leaf structure or any other morphological characteristic that's important for identification. And then, of course, a lot of ID comes down to the flower or reproductive part. So ideally, your plant specimen would have either flowers or fruit. It will then also have a label. Generally, they're always in the bottom right, and that label is going to have the metadata or the data of your data, which is, where it was collected, when it was collected. Often it'll include what other plants are growing with it. But especially some earlier plant specimens, they are very minimal on the metadata. The last component of a herbarium specimen that is important to prioritize is the use of archival materials. When well cared for, herbaria specimens can last hundreds of years. We still, by we, I mean some very important herbarium elsewhere, still has, Linnaeus's original herbarium specimens. So especially if they're well cared for and preserved using acid-free paper and acid-free glue, they can be shelf stable for a long time and serve as that, data source for decades to come. So metadata is the next thing I wanted to mention. So as I said, some specimens are scant on data, but when you are, say, making a specimen today, you want to make sure that you include as much data as you can that gives us a little sneak peek into where that plant and how that plant was growing. So of course, it's going to include the location information, what county you're in, what state, what the plant was identified as, but then it's going to have specific location information with coordinates, ideally, and then also a description of the habitat. So here it's in Blountstown, but the habitat is there on a field boundary on the long side of a road. So that's really important. And when we're digitizing information, which I'll talk about later, the discrepancy between the location and the habitat is sometimes a tricky one. But you'll see here, it'll also include a description of the plant, any characteristic that can't be captured on the specimen. So here on the label, they include that the tree was up to 20 meters tall, since that isn't something that the specimen can reflect. It'll also give a little bit of a leaf description there, and often it will include associated species that are also growing with it. So here's that same beautiful yellow lady slipper that I showed earlier. Again, this is just an example of a label that, before standards were implemented, this is pretty scant on data, but it's still some important information that we can glean. And again, it's that hard physical reference that it was there. Now I want to talk a little bit about the artistry of these specimens and really just kind of call out the intricacy that can go into caring for some of these really delicate features. This specimen here that jump preserved, again, this was collected back in 1940. I wish you could see it in person, but this beautiful lady slipper pouch is actually filled with cotton. So they filled that cotton to help preserve the 3D nature of the flower pouch there that's so characteristic of the Cypripediums, which I thought was just a really, wonderful example of the artistry that can go into herbarium curation. Just another example of that, I love this specimen of Delphinium exaltatum. This is a globally rare plant. This specimen is from North Carolina. And this is just a perfect example of the care that goes into making these 3D specimens 2D, yet still reflect the 3D nature. Especially with these really complex flowers. This is within the Ranunculaceae, so we have big spurs that are characteristic of that family. And so having, this is just a wonderful example of that beautiful spur and this other wings and flower characteristics. So as you would imagine, large herbarium collections can yield, a lot of really beautiful specimens, really beautiful works of art, very important data, often several type specimens, and also a large network and platform that allows them to share data with other herbaria within their networks. So some good examples of these kind of classic large herbarium institutions include Kew Gardens. Kew Botanical Gardens also is really well known for their research department, and they have a multi-floor herbarium where they do much taxonomic work. Another example of this is the New York Botanical Garden it's a huge repository of specimens and is also kind of an international focal point for trade and loans and other conversations between herbaria. And I'll be circling back to the New York Botanical Garden Herbarium again. So these large herbaria, right, obviously important repositories for data and a very important regional and international institution. But this is where I want to shift the focus to the significance of small herbaria. And small herbaria, while they, have fewer specimens and cover often a smaller geographic range, they're really important because they help fill data gaps within local or regional floras. They also can help track climate change at a local region, at a local scale, because of that metadata. If we have on the plant labels when they were flowering or when they were fruiting, we can actually track over time how flowering time within a region may have shifted or changed in response to some kind of environmental shift. They're also really important for documenting local expansions and also local invasions, invasive species. We can often document when these species spread throughout a region based on their prevalence within the herbarium record. We can also use small herbaria to identify patterns of biodiversity concentration and use that to really highlight some of our local biodiversity hotspots. And then the last thing that I have found to be really special about small plant collections is the glimpse that it allows us to have into the legacies and personal lives of some of our most prolific plant collectors. Which leads me to the herbarium at Frostburg State University. So I was delighted to find out that we had a herbarium collection at Frostburg when I first started teaching full-time in 2021. The herbarium is right there in Compton Hall in room 317, and it was filled with about 15 cabinets of archival specimens, anywhere from 2000 and earlier, plus 3 cabinets of teaching and student specimens, and lots of unmounted specimens, many of them still in newspaper. So kind of what I had gathered is that our herbarium was in excellent shape, but it hadn't really been reinvigorated for a while. And this was exciting to me because I did a little bit of herbarium work in undergrad at JMU, and I really liked creating and working with plant specimens. So the first thing that I did was get us registered with the New York Botanical Garden. At NYBG, they have the Linda Steer Herbarium, and also within that, what they call the Index Herbariorum, which is essentially an online database of other herbaria within this network. So once you're established within a herbarium network, this allows you to network and communicate with other herbarium curators. And again, this is especially important for any taxonomic research. Typically, when researching a species, you would have to request loans and plant specimens would have to be mailed. And that was, a kind of cumbersome process, but not getting registered, but mailing loans. But now that we're registered with New York Botanical Garden, you can actually search our herbarium. You can either search Frostburg State University or our official herbarium code, which is FSUH. So now that we are within this repository, we're now connected with this larger repository of online and modern herbaria and biocollections. So worldwide, we have over 3,000 documented institutions of registered herbaria. And again, this includes international collections all the way down to just personal private collections. So these are registered collections that are within this greater network where New York Botanical Garden is connected to. So then from there, I got involved with kind of a larger approach or more regional approach to digitizing bio collections through a large nonprofit called iDigBio. So this program essentially works on digitizing all kinds of bio collections: fishes, mammals, insects, fossils, plants, the whole gambit of biological specimens. And within iDigBio, we have different taxonomic levels, so, plant world. And then within regions, we also have these, we have these other subgroups within regions. So within that iDigBio network of biorepository, we also have this whole inner network of regional herbaria within the country. So you may have heard of CNET, they're really big out west, or most likely CERNEC, which is that's the southeastern United States kind of regional chapter of herbaria. But Maryland is actually included in the Mid-Atlantic Herbarium Consortium, which includes Maryland and Northeast. So from there, we started to connect with the Mid-Atlantic Herbarium Consortium, or MAHC. And this is run by an acquaintance that I'd actually met from fieldwork a few years prior. So it was really fun to work with Cindy Schema again, but Dr. Cynthia Schema is the research botanist at the Morris Arboretum in Philadelphia, and she is also one of the main champions of the Mid-Atlantic Herbarium Consortium. So I reached out to her about how can we get our collection digitized and part of this larger online biorepository. And she helped teach me about kind of the steps of digitizing a collection, which pretty much you have to go through all your specimens and make sure they are in good shape. You have to fix them up, repair them, glue, add fragment packets to retain any loose materials. And then you have to fix up or prepare the specimen for imaging, which includes a little measuring scale and also a color code. It then gets an official barcode with your herbarium code and a unique identification number so that it's scannable and searchable. And then it gets imaged. And I'll talk more about that imaging here in a second. The data will then get transcribed and uploaded, and then you still have to design and make your database. And that is what goes into making an online digital herbarium. So, that's a lot of steps, and I was a little overwhelmed, but I was really, really fortunate that my friend Cindy said, hey, bring your collection to us. It's small enough. We will digitize it for you. So that was so amazing, such an amazing offer from our friends at Morris Arboretum that last spring, or this was actually 2 springs ago, I, with the help of a few colleagues and students, packed up our whole herbarium, took about 73 specimen boxes. It was a lot. And we packed up a university van and drove out to Philadelphia and dropped off our specimens for digitization. So the imaging portion, this is Lulu Korzak. She was another integral component or player there at MAHC that really helped us get our specimens digitized. And she explained to us the imaging process, which essentially you have this imaging box, which I learned is actually the same device that people will use to take nice photos of, any goods that they're selling online. So that nice, necklace that you saw on Etsy, the photo was probably taken in this nice photo box to take a, really bright and well-focused photo. So we have this bright light box, and you'll see up in the left corner, you can see the camera rig stationed up above, pointing down. And then the image on the right shows the software that then shows the photo that was taken, and you can then, accept it or, retake the photo. And again, I just want to give a huge shout out to MAHC for doing this work for us. It wouldn't have been able to get done otherwise, and that work is very much appreciated. So the fruits of their very, very hard labor has resulted in FSUH officially being on MAHC, or that Maine Mid-Atlantic Herbarium Consortium website. When you log in there and search collections, you can there see Frostburg State University there. And again, this consortium includes any herbaria within the Northeast, and it includes Maryland and up. So that includes other Maryland universities, as well as the Maryland Division of Natural Resources. Their herbarium is also registered on MAHC. So now that we're here online, we can also see a really nice kind of summary of our collection there. You can see the collection statistics there. That's the page that it would show online. That's a little hard to read, so let me show you some figures of it that one of my students made to represent the results from digitizing our collection. So in terms of taxonomic diversity within our collection, we can see we have 184 plant families, 976 genera, 2,257 species, and then if you include varieties and subspecies, we have 2,396 interspecific taxa. We can also see that the collection is almost totally angiosperms, or flowering plants. We have over 15,000 specimens of flowering plants, only 240 of gymnosperms, and 337 of pteridophytes, or ferns and fern allies. Also at the bottom, you can see the most common families that we had specimens of. And this actually really nicely aligns with the families that are also most diverse in our region. The asters, roses, mints, mustards are all, some of the most diverse families in our region. So that kind of really nicely supports that we also have the most specimens of those families too. And then in terms of regional results, my student also looked at how our specimens were concentrated throughout the state. So you can see we had over 15,000 specimens of the United States, but we also had a number of specimens from other countries. And then within the United States, as you would expect, the majority of them are from Maryland. But we also have many specimens from West Virginia and North Carolina and Illinois, which there is some fun background into that as well. So also within the state of Maryland, we have a map that also then shows the concentration of specimens by county. So again, as you would expect, we have the most specimens concentrated from Garrett and Allegheny counties, which almost certainly is a product of student plant collection projects at FSU and students doing many of their collections within the region, or back home when they go home for breaks. We can also see a higher concentration of collections made from adjacent West Virginia and Pennsylvania, Somerset County and Mineral County, and then down into Grant County and West Virginia as well. And that's believed to align with the large swaths of public land in those regions, many state game lands to the north in Pennsylvania, and many forests and natural areas south of us into West Virginia. We also had many specific collectors that had certain places they like to hang out, which we'll get into again a little bit later, which is also attributing to these different state results. So those are the results in terms of the diversity of our collection, but in terms of accessibility, the coolest part about being digitized is that anyone could go on to the MAHC site and search either by taxon, right? Say I want to look at all the specimens for Elisma subcordatum and put that in and look at those nice pictures. Or you could search by institution, just by Frostburg specimens. Or my favorite part, you could also search by collector or by date or by location. So having these specimens online with the data fully transcribed opens up so many doors in terms of data accessibility and ability to quantify this data at a much faster rate than imagine having to request mailed loans of every single one of these species, when now we can just get online and look at the label ourselves. One of the other really nice features to this is on the backside, in terms of being a curator. When it comes to being able to edit or transcribe our specimens, on the back end, I'm able to do batch edits and batch annotations, which is really, really helpful, for example, for name updates. So instead of going through every single one of our, tree club mosses, which is now Dendrolycopodium, which was Lycopodium, right? Now I can select all of that exact same species and do a batch annotation update instead of physically annotating every single one of those specimens. So it's really opening doors for curation as well. So now to jump into some of the what I think are some of the cool finds of our collection, that kind of was a product of this digitization effort, was seeing species that were believed to have been locally extirpated. One example is Blophelia hirsuta, or hairy downy mint, or hairy wood mint. So this is related to Blophelia ciliata, which is the downy wood mint, which is more common in Allegheny counties. But here we had a record only in Garrett County of this hairy wood mint. And I looked online at the flora of the southeastern United States and saw that in Maryland, it once occurred, but is considered historic. And I thought that was really interesting. I also thought it was interesting that this was collected by Melvin Brown, who I'm going to circle back to again later. So it actually is interesting that I, happened to stumble across this specimen as I was sorting things, because just last year, it got me thinking, I wonder if this specimen is still around. And I jumped on iNaturalist and did a little iNaturalist search and saw that one of my acquaintances out in Garrett County had actually observed one a few years back outside of Grantsville, like this label had suggested. And last year, I went out myself to that same site and was able to re-find that Blophelia hirsuta, and see it with my own eyes and make a new collection, a new plant specimen to, really document that official rediscovery of that plant in Western Maryland. So that was pretty cool. Some other cool things that have come out of digitization is actually looking more at the legacies of certain collectors. And there are two really prolific collectors that I want to highlight tonight. And the first one being Franklin C. Lane. So if any of you are familiar with Frostburg State University's campus, you have probably been by or been into the Lane Center. This is kind of the student hub of campus. It's the hangout spot. It's where the students go to get food and relax. And this building was actually named after Franklin Lane. So Dr. Lane came to Frostburg in 1960, and he eventually became the department chair of the biology department. And then just 2 years later became the dean of students. He was extremely well liked, and many of his mentees, have remarked online, since his death, how much, of an inspiration he was to them, which is really cool that he wound up at Frostburg, because he actually is, he did much of his work in Wisconsin. He went to Northland College, which is on the border of Lake Superior. And there he studied plants. And after he graduated for his master's project, he was actually contracted to do some of the first plant inventories or surveys of the Apostle Islands in the Great Lakes, which just sounds like a botanist's dream, to be the first explorer of these amazing geological and biogeographic relics. So what was really amazing to find was that much of Franklin Lane's collection from those initial Apostle Island surveys are in our collection. which is just amazing to see some of these specimens that you can see were collected in the 50s, still holding up over time, and really just being such a cool cultural and historical resource, as well as these ecological data resources. So Franklin Lane, other than being, a botanist checking out these cool places in Wisconsin, and then he comes to Frostburg and he's really well liked and he's a great teacher, he also was an orchid guy. And he has many, many beautiful orchid specimens that are in our collection. And I feel so blessed to have them here, to be able to see them firsthand. Because again, Franklin Lane was one of those people who took great care to really make sure that the specimen was honored. If you're going to collect a plant and kill it for these research reasons, he really made sure that it was perfectly positioned to serve its maximum use in the herbarium. So you can really see that beautifully here with another lady slipper orchid. We can see that again here with a coral root he collected on the Apostle Islands. He really liked coral roots. He collected a lot of Corallarhiza. And if you've never seen that in person, here's an example of the same species, Corallarhiza maculata, that I saw. The right is a photo I took, and on the left is another one of Lane's specimens. Again, just really depicting the intricate details that you can still glean from these specimens, these 2D specimens, despite losing that original 3D shape. So Franklin Lane's collection is definitely a really cool component of the herbarium that, again, I hope to show off more in the future and investigate a little bit more of his background and where he liked to botanize in the area. The next botanist that I want to highlight that we have learned a little bit more about through digitization is Dr. Melvin Brown. Now, in contrast to Lane, Dr. Brown was a local. He was from Mineral County, West Virginia, just south of Frostburg. He went to Potomac State University, and then he went to WVU for both his master's and PhD. He was a student of Earl Core, who you may know from the Earl Core Arboretum at WVU. And he also, Dr. Brown, was really well known for his own personal arboretum. He was more of a dendrologist, I would say, really, really, really into tree research and grafting and horticulture. So other than his own personal arboretum, he also established the Kitzmiller Botanical Garden in Kitzmiller, Maryland, out in Garrett County. And he also had given many, many donations to other arboreta that used his original specimens as some of their botanical collections. So this includes many plants at the WVU Arboretum. Also, the James Madison University Arboretum also has legacy trees that came from Dr. Brown's personal arboretum, as well as the Frostburg Arboretum. So Dr. Melvin Brown, though, despite, he's really into trees, he's a tree guy, but really what he is most well known for is actually his major contributions to Maryland botany, aside to his work at Frostburg. So in 1972, he actually published the first woody plant inventory of Maryland, the woody plants of Maryland. And then in 1984, he published another similar manual of the herbaceous plants of Maryland. And these are the only, up until in 2021 with that new flora, these continue to serve as the primary regional floras and guides in our region for Maryland plants until the publication of the Flora of Virginia in the early 2010s. So this, for a long time, these publications served as, a really important reference for regional botanists, not just Maryland botanists as well. And so Dr. Brown's legacy at Frostburg is a cool one, but it's kind of unknown, and it's something that I look forward to investigating further. But what I can say is that he also left behind some really wonderful living and biocollection legacies, including both living collections in our arboretum, which includes this beautiful mature bald cypress in the Frostburg State University Arboretum. And many of you know, like, hey, bald cypress doesn't grow in Allegheny County. But that is because this was one of the specimens that Melvin Brown first planted when he started working at Frostburg. And now it's a beautiful, wonderful, mature, gorgeous bald cypress that turns beautiful rusty orange in the fall. And I teach and use this specimen to teach my students every year that species. And it's really special to me that I get to teach that from, using that specimen, the same specimen that Dr. Brown used for the same classes 50 years ago. So that's really special. In addition to his living specimens that he left behind, he also was a major contributor to the plant, to the herbarium collection. I mentioned earlier that Blophelia, or that wood mint species, was a collection of Dr. Brown's, as well as some other really important regional specimens, including this one on the right, which is Ripariosida hermaphrodita, or Virginia mallow. And that actually leads me to some of our current initiatives that the herbarium is being used for, other than, those investigations into Lane and Brown's personal lives. We can use these specimens for lots of different projects, and primarily we're looking at conservation. So several years back, actually Cindy Schema, the director of the Mid-Atlantic Herbarium Consortium, I first met her a couple of years before my herbarium venture, assisting with field work, doing surveys for the globally rare Virginia mallow. And this plant has a really weird spotty distribution within the East, but it was historically known from several counties along the Potomac River. I assisted with some of those surveys, but she went and personally surveyed every historical location she was able to glean from herbarium specimens and other data sources. And unfortunately, she found that most of the populations had been wiped out. They're very easy to succumb to competition from invasive species. However, the only extant population of this species in Maryland that she was able to find was at the same location as Dr. Brown's herbarium specimen for it. And I've been able to see the population myself. And that is just so cool and so special that we can glean this information from these historic records and use that as a way to track and kind of inform these other conservation initiatives. That study was primarily looking at genetic variation and speciation within that spotty distribution. We can do similar studies. We recently helped a study with Pennsylvania DCNR, doing a similar study with another globally rare plant, the bog Jacob's ladder. This is another specimen. They reached out to me for assistance with our Maryland populations for collecting genetic material. And I was able to pull out some of these physical herbarium specimens and figure out where we should sample the plants for their genetic study. And I was able to get that collection permit and help them with that, which was, again, a really fun and special collaboration. In addition to those conservation initiatives, we also use the herbarium for undergraduate research. I've taken 2 students to conferences where they were able to present on the things they researched regarding the herbarium collection. My student Kayla on the left did a really intensive search for all of the specimens in our collection of rare or threatened species and was able to kind of identify areas for rare plant conservation prioritization. And then my student Lily on the right made all those beautiful figures and maps that summarized the data from our digitization initiatives. So these are just a couple of examples of how the students can learn, these hands-on plant curation skills, but also learning how we can glean and use that data to inform conservation. But primarily, the herbarium specimen collection at Frostburg is used to teach. We do have a relatively large teaching collection that is primarily made from student collections. Oftentimes, the plant classes incorporated some kind of plant collection project, so I was happy to include that when I started teaching plant systematics as well as dendrology. So on the left, my students are looking at the specimens during a lab practical, but also on the right, you can see the students preparing their own specimens from their collection project. So that has been a really wonderful teaching and learning resource for them. I will say that our teaching collection did have a lot of holes in it, and our beautiful archival collection, while those specimens can be used for teaching, as you saw, many of them were very old and very delicate and really should not see the light of day to really keep them preserved. So I didn't want to teach on those. So one of the most, the biggest initiative that I've been working on the last couple of years is just trying to fill in some of the gaps in the teaching collection. So now I'm just going to show you some pretty photos of specimens that we've added in the last couple of years to the teaching collection. And I don't know what it is about ferns that scares people, but we have no ferns in our collection, even in our main archive, not just the teaching collection. So the last several years, I've been trying to opportunistically collect some ferns to fill in those gaps. You can see on the left is our common polypody fern. And on the right is probably the biggest rattlesnake fern I've ever seen in my life. But it was a beautiful specimen there with its spores on that fertile spike there. Continuing with some fern appreciation, some beautiful maidenhair ferns, as well as some walking fern, one of my personal favorites. Another example of some holes that we had were invasive species. And I mentioned earlier that invasive species can really help us, or collections of invasives can help us track them. But I noticed in the teaching collection, we were really missing a lot of common things, even just things like bitter or climbing nightshade. This was growing right outside of Compton Hall on campus. And I saw the beautiful flowers and said, hey, let's make a specimen of that. So trying to just opportunistically fill those holes when I can of non-native things that were also lacking in the collection. And then the other area of the collection that is very underrepresented are any species that flower later in the year, because a lot of those student collection projects are in the spring. And as you know, in Western Maryland, we're pretty delayed with our spring wildflowers. We really don't get our spring ephemerals until late April. And when classes wrap up just a couple weeks later. So we really were having a huge deficit of some of our later flowering species. So I also have been trying to put more efforts towards some of those summer plants, like our beautiful cardinal flower on the right. Or also, as you would guess, people don't like grasses. So grasses were also really underrepresented in the teaching collection. So we've been doing a lot of collection of those as well. So if you guys are ever interested in looking at these specimens from Frostburg or from any other herbarium, you can jump online, either go to the Mid-Atlantic Herbarium Consortium or any other of those regional repositories, or you can even go straight to New York Botanical Garden or Kew and search their online repository as well. The photos are taken in really high quality, so you're able to mega zoom in both to the label and the flowers, and it's honestly really fun to just kind of peruse those images sometimes. So the last thing that I want to talk about is actually just some challenges that herbaria, especially small herbaria, are facing. You may have heard last year or the year before that Duke University decided to close its herbarium, which is one of the most well-funded and most recognized colleges in the East. And, if Duke can't even find a way to fund their herbarium, that's a really kind of glim outlook for some of our smaller herbaria. But despite that, a petition was signed, and I don't know if they're still totally saving, but I think some kind of agreement was made to keep a part of the collection on campus and diffuse the remainder of it. I think it was a space holding problem. So, those are just some challenges. Funding for that is one problem. Another problem too is that herbarium curators are, it's a full-time job, but most herbarium curators do it either part-time or free. This is what I'm doing just because I love the collection, but also many of my colleagues that I talk to, other herbarium curators, they're professors and have a normal teaching load, and they just do it because because it's an important resource. So it is something to recognize. It's a challenge to Frostburg's herbarium and other small herbaria. But I think especially in light of climate change and other environmental challenges, the importance of these biocollections are really shining through and giving us a reason to give more focus on our small plant collections. So that is it that I have for this evening, wrapping up a little bit early, but I want to say thank you again to Maryland Native Plant Society for having me chat this evening, and thank you again to you all for tuning in for a brief exploration of the plant collection at Frostburg. And with that, I would be happy to take any questions.
Thank you very much, Clara. That was very interesting.
Ben, you can go ahead and unmute.
So Clara, noticed that you had mentioned a name change and someone else answered the question I had for the Lycopodium for the new name, and I appreciate that. I think her name was Margaret. And then the other question I had was, for a name change to officially happen, what's the process for that? And like, what's the official body that would, I guess, make that change within, whether it's a county, a state, or the country, you know, just to say this is the new name. And one of the examples that I was thinking of was kind of recently was rosemary. And although it's not a native, you know, I know it got moved from rosmarinus to salvia. And I still encounter people that still call it Rosmarinus officinalis, but it's now Salvia rosmarinus, I believe. So if I'm wrong about that part, but correct me. But I just wanted to figure out how the name change is officially changed. Thank you.
Yeah, Ben, that's a great question. That's a really good question. So there is actually an international group of botanists that they're part of the International Code of Botanical Nomenclature, and they meet annually somewhere abroad every year, and taxonomists from all over the world come to these meetings. And essentially they get together and they make and adjust, you know, the official rules for plant naming, plant taxonomy. There is a similar group or code for zoological naming, but I will note that they are completely distinct. And that is important to note because it has happened. Technically, plants and animals can have the same scientific name because they follow different rules, but that's a story for another day. Anyways, the botanical code essentially has these rules, and one of the major rules it says is that plant names are retroactive, meaning that they recognize the first officially published name. So the name that was the first one to follow a proper publication route in a peer-reviewed journal or book. So what happens a lot with these name updates is that a plant was described by two different botanists in two different regions where communication was not really easy. And authorship was given to one, but then later it was discovered that the other author actually published it before. So a lot of these name changes are because of better understanding of the actual authorship. Now, the other time this happens is when we actually have more biological information or evolutionary evidence that suggests that a species is more closely related to something else. So I am not a mint expert, but my guess is that some kind of molecular analysis was done and was actually able to track the genes of these plants to show that rosemary was actually more closely related to salvia than what would be deemed being its own species. And that is happening a lot, especially I mentioned at the beginning, species description or species discovery is still happening, you know, pretty frequently. And most of the time it's from molecular or genetic studies. And that is kind of resulting in either a name change or the description of a new species. I hope that answered your question.
Michael Wilpers, if you can go ahead and unmute and ask your question.
Well, just first, I have a friend from high school who graduated from Frostburg State back in 1977. So I have a small connection. And I was very happy to attend the Maryland Native Plant Society conference there a few years ago. My question is about the Browns and their collecting and the flora. Do you have a sense from from the collection and your knowledge whether the plan to create the woody plants of Maryland or the herbaceous plants of Maryland stimulated a lot of collecting of specimens that wouldn't have happened otherwise, and whether he was in touch with botanists, field botanists from all around the state, saying we've got this deadline and we want to put out this flora and we need a lot more specimens in this, that, or the other area. Do plans to, in general, to produce flora, are they great stimulants to collecting and adding to herbaria?
Absolutely. So to do an official floral inventory, I mentioned earlier that herbarium specimens are that solid proof of documentation that are so important to publishing floras today. So having that physical reference that you documented this species in this county, especially if you're doing a state flora, having that county documentation is really important. So I'm sure that a lot of his collections, like you said, were probably definitely in part to his research for his publications. Something else that I've noticed about Dr. Brown's collections is that it's kind of fun to be able to trace their old haunts. I can tell where he liked to go botanize. He liked to go up to Dolly Sods. He liked to go back home to Mineral County. And you can see his concentration of collections around, where other botanists in the region like to hang out, like Fenzel Swamp and Dolly Sods especially. But yeah, definitely it's interesting to be able to trace the personal goals or collection projects of an individual just by their specimens. It's a really unique kind of human dimensions component to the bio collections.
Joyce asks in the chat, how is the decision made as to what specimens are included in a herbarium collection?
That's a great question. And really it boils down to the discretion of the curator. So one of the, cardinal rules of a herbarium is to never throw anything away, throw away no plant parts ever. But also there's a certain point where a herbarium only has so much space and I don't need, my 800th student collection of Christmas fern. I don't need to keep those. So it is just up to the discretion of the curator. Some herbaria, keep everything, every single specimen. But what often happens if there's, extra specimens that a herbarium doesn't need necessarily, what they'll often do is loan it out and give it to another either nearby herbarium or a smaller herbarium that could use some extra specimens. Also, some of them often get donated to local high schools or envirothon teams. They can serve as, learning tools for lots of different places. So often what herbaria will do will donate them to other institutions if they have too much.
Nikki asks, can you talk more about those human connections? Why were those other states so prominent in the collection?
Yeah, let's see. So Illinois was a really popular one, or a very specimen heavy one. And that was actually because Dr. Lane taught in Illinois before coming to Frostburg. So he did a lot of collecting there and brought that with him. We also had a ton of specimens from North Carolina. That was another high concentration. And there were two other botanists that I wanted to talk about, but this talk would have gone way too long. So I had to cut them out. But there were two other prominent botanists, A.E. Radford and A.H. Ailes. And both of them were North Carolina botanists. They worked at Chapel Hill. And they worked together to actually write a textbook, the Handbook of Vascular Plants. I had a slide that had the cover on it and everything. And they also did a lot of taxonomic work in the Carolinas. Now, how we wound up with their collection, I have no idea. And so this is, what's really exciting about diving into these, when I said the legacies of these collectors, it makes me wonder, did the folks at Chapel Hill have duplicates and they sent us their extras? Or did we have faculty come there to Frostburg like Lane did and bring his personal collection? I don't know. I'm still trying to piece the puzzle pieces together, and it's really fun to try to figure out what these people were doing and why they wound up in Western Maryland.
Lauren, I see you have your hand up. You can go ahead and unmute.
Yeah, thanks. This is quite fascinating, actually. So I'm wondering, are you able to use these specimens as to extract DNA ever? Has that ever come up? Do people, I think it probably does, but I'm wondering how often, or what are the challenges there, and how do they hold up over time?
Yeah, so I am not a microbiologist, and I personally have not, or probably will never dabble in genetic analyses myself. However, I know many taxonomists who are using genetic analyses, either partnering with other labs to do that analysis for them, or looking, for some kind of molecular component to really try to trace the evolutionary histories of some of these species. So yes, they are collecting live tissue if they can for many of these studies, but people are frequently and commonly using these herbarium specimens as a source for genetic tissue. Now, some methods are really destructive and, require a lot of tissue. But with our more modern applications, you can get away with using just 1 square centimeter of tissue. So, for example, the Virginia mallow specimen that I mentioned was used to refine the specimen in Allegheny County. They also took a sample of my specimen there from Dr. Brown, and they were able to use that in their analysis. So it's really special, that our collection can help in these regional analyses. And kind of, again, it's just like a puzzle piece trying to figure out the evolutionary history of some of these taxa. And it's, the special location of Frostburg is what allows us to have these special specimens that can be used for this really cool research.
Thanks. I'm sort of thinking about it as a possibility if we get to the point where we can actually identify ecotype level, groups. Those collections will be crucial, really.
Yeah, well, they're able to. You're with the genetic analysis, you're able to identify unique subpopulations within the metapopulation that is, as you would imagine, integral to conservation biology for rare species, but definitely has applications for, seed growth and local ecotype gardening and all that as well. That's a really good idea.
Steve Lonker, I saw you had your hand up. Did you want to unmute and ask your question?
Yes, I have 2 questions here. Has anybody in the past or even in the present been collecting ecological data? Imagine seeing how the environmental landscapes have changed since the 18th and 19th century. And the second question is, what's the status of machine learning algorithms for both species identification and phenotyping? We're talking about AI here.
Yeah, okay, so yes and yes. So for the first question, ecological monitoring. Yes, a lot of different institutions are engaging in some kind of ecological monitoring. A lot of places are either doing vegetation monitoring or soils, atmospheric testing, or phenology using a phenocam to look at changes in when plants leaf out and flower, etc. At Frostburg, we do some long-term monitoring. We have a research forest plot where we do vegetation monitoring and soil sampling every year, and other institutions do that too. But I would say the largest is, the USDA has, a veg map essentially of the whole country of the generalized vegetation cover. There are other institutions that monitor and collect that data, and many of them are part of these larger scale data sharing. So there is a way. What I think you're asking is, yes, we have this ecological data that we have access to that we can then align our herbarium data up with. Yeah. So when we see changes in phenology or changes in plant associations, on that herbarium label, we can often align that with, this weather data or this other ecological data, which is really fascinating.
Yeah, a question I was referring to anything in the older collections as well. And I was getting into the machine learning algorithms. Very curious about that.
Yeah. So then in terms of AI, yes, machine learning has become a popular tool, especially with herbarium biocollections. And I'll give you a really cool example of how this can be used. The Duke Herbarium actually recently put out a research study where they use machine learning to look at herbarium specimens, and they were able to, with much more confidence than humans, identify different plant pathogens from the plant specimens.
Wow.
So just on the plant specimen, they were then able to glean that same location information as parasites, bacteria, insects, all these different, galls, all of these other, pieces of information that we can find on a plant specimen. Machine learning and AI has been able to detect that and identify it as additional observations, which is really neat.
Thanks.
So I don't think we have any more questions in the chat. We do have a number of comments. Interesting presentation, wonderful presentation, really nice presentation, wonderful talk. Dominic has the additional comment that he remembers that he enjoyed preparing herbarium specimens at Frostburg in 1992.
That's awesome. Dominic, do you remember who your professor was? Was it Linda Lyon? How you worked with the Morris Arboretum. Yeah, it was so cool how that kind of came full circle and the folks there were able to help. And again, I cannot underscore how major of an initiative, that was for them to undertake and help us with that digitization. So that was just super awesome. Any last call for questions?
Donna Ford Orange says Sunshine Brosi, if I pronounced her name correctly.
She was my predecessor. But she wasn't there in 92. So, but yeah, I'm not sure. I can't remember who the botanist was in 92, but I am glad you had fond memories of plant collection. I hope my students feel the same way in a couple months.
Dominique says Simon Dabadine.
Haven't heard of them, but they must have been a wonderful teacher for you to remember it. From leaves to labels.
Chris S. Do you want to unmute yourself? You just put a comment in.
I have some background with machine learning, and I put some articles about applying machine learning to herbaria. This particular one appears to be focused around image recognition and leaf machine 2. I just thought that might enrich the discussion.
And specifically, it says the article is From Leaves to Labels, Building Modular Machine Learning Networks for Rapid Herbarium Specimen Analysis with Leaf Machine 2. And if people are interested, Chris has provided a link there.
All right. Well, thank you guys again so much for having me this evening. And I look forward to chatting with you guys more, seeing you guys on a plant walk, hopefully sometime in the spring. Stay warm out there.
Yes. Thank you so much for joining us. And again, everyone who's participated, thank you for coming and asking great questions.